mentioned in my title, my original title for the talk, we develop a lot of tools. I'm going to tell you a little bit about our work in the lab, and also I'm not going to talk specifically about Sweet2P because I don't know if it's relevant to everyone here, but I'm going to talk about like some metrics that we use to benchmark if data is high quality or not, because I think that's useful as data analysts. And I'll talk about our research as well. So we're interested in understanding visual processing, and so the visual world can be complex. Everyone can hear me okay? Okay, cool. So this is where's Waldo? Does anyone know, can anyone see where Waldo is? I mean, it's Friday morning on the last day, so. And I chose a hard one as well. I get extra minutes at the end, Ben. No, I'm just kidding. So I'll point him out. He's here. That's working. Your visual memory system is flawed. No, I'm kidding. Well, not flawed. It's yeah. Actually, we'll talk a little bit about visual memory too. So, when you try to do this task, you might have been imagining Waldo and looking for like features like his glasses, or doing texture recognition, looking for his striped shirt. Which in this one you can't even see his striped shirt, so it's like really hard. And while you were doing this task, for those of you who've done Where's Waldo before, you have a memory of what Waldo looks like, which is a compressed representation of Waldo. And so that's something we're also interested in studying in the lab. So, and while you did this task as well, you were integrating your visual experience with your motor movements and saccading around the image. And so when we think about visual processing, we're thinking about a lot of these different steps. And so to study these questions, we use 2-photon calcium imaging. So here's an example recording from mouse V1, where we use GCamp8S, which we're lucky to have here at Janelia. It was developed here at Janelia. Now it's publicly available, this mouse line. And so you can see these cells lighting up when we show images. The big advantage of GCamp8S is it's very fast, and so we can show images at a 7.5 hertz presentation rate. So this is actually 2 times slower than the presentation rate the mice see. And so we show up to 65,000 images in a single imaging session, which means we get a lot of data, which is great for training deep neural network models of visual responses. We also do large-scale recordings in the lab with GCamp6, or more recently also with GCamp8, where we use a large field of view microscope, this mesoscope, which allows us to capture over 4 millimeters by 4 millimeters field of view. And we do temporal multiplexing, so the laser pulses go one to this higher depth and every other one to the lower depth. So you get this plane, this plane, this plane, this plane. So we get 2 planes basically simultaneously, which gets us on average around 50,000 neurons at a rate of 3 hertz. And so this kind of imaging is great for kind of surveys of what's going on during various behavioral tasks. So we can get primary visual cortex in mice, this V1 area, and all these higher order areas simultaneously. So we use Sweet2P, our processing pipeline, to detect these neurons automatically, because you don't want to circle all of these neurons. So we get these masks that look like this, and then we average the pixels in each mask on each time frame of the video, and that gives us these example neuron traces. And so I'll tell you briefly about Sweet2P and some metrics we use for benchmarking calcium imaging data. So this is what your raw data might look like. So we're doing these recordings in vivo. The mice are free to run, whisk, do whatever they want, so you see some motion. So the first step you always want to do is motion correction, because as you can see the uncorrected video, in order to get a good trace of activity, true activity, you need to make sure that that cell is not jiggling around, because then it might look like activity when really it's movement. And then we do ROI detection, looking for correlated pixels across time, so using functional information to do ROI detection. And that usually works better, especially for high SNR recordings, although we do have an option to use our anatomical algorithm CellPose inside Sweet2P to do anatomical ROI detection, which is useful, especially for lower SNR recordings. And then from there, we do signal extraction, what I was saying, summing pixels, and I'll talk a little bit about neuropills correction as well, and then deconvolution. And then we output your files in MPY, MAT, or NWB format, which is bolded here. So you can upload your data almost straight to Dandy from here, although Ben can correct me if I'm wrong. So you can easily share your data with the community after you process it through Sweet2P, and you can use our GUI to also explore your data and to curate your data. Okay, so Sweet2P runs pretty fast. So these are benchmarks on Google Colab. For a short recording of 4,500 frames, it should run in less than 10 minutes, basically. Okay. Are there any questions about that? All right. So the first thing I was saying was that you want to make sure that your the calcium traces that you get are true activity traces and not related to the motion of the brain, potentially. So the way that we quantify motion in the Sweet2P GUI, and you can also do it outside the GUI, is we take the movie, which is X by Y by time, and we take the principal components of this movie. We usually take a subset of frames. We'll take like every 10th frame or something like that, and we'll take these principal components and look at them. So this is, for example, the top principal component of the movie across time, and we can look at the average frame from the top frames here, basically, of this PC, and then the bottom values of this PC. And so these are like the top. This is the average of when the PC is highest and when the PC is lowest. And then we can look at this difference image. And in the GUI, we actually have this option where you can kind of flip back and forth between the top and the bottom image. And for those of you who do calcium imaging, you might look at this and try to look for motion, and it might be, hopefully it's clear, this doesn't look like a principal component that's related to motion. It looks like it's just activity. So you see these cells that are lighting up that weren't there before. If there's motion, you'll see kind of like jiggling parts of the field of view, and those will sometimes come out. Or you'll see different things like cells coming in and out of the field of view if it happens to be Z motion. So we find this really useful to make sure. And so if you do find a PC, for example, that's related to motion, you might want to make sure that variable is not correlated to other behavioral variables, for example, in your recording, to make sure that that's not driving some of the effects that you see. So we especially try to be careful about that, as you'll see when Atika presents, that we're relating behaviors to neural activity, and we want to make sure that it's not related to any movement of the brain or anything like that. So what you can do is you can take basically do a registration between the top frame and the bottom frame and look at the registration offset. And ideally, it's quite low. So in this case, the rigid offset is 0, so that's good. And the non-rigid max offset, so kind of like this piecewise rigid registration that we do, is also quite low, below 1 pixel, even below 0.5 pixels. So it seems like we've corrected this movie well. Does that kind of make sense? And what's nice with the Dandy sets, so some of them do have raw activity, so you can just download their raw data and apply this technique if you're worried about some of these artifacts. So another thing to think about when you're looking at calcium imaging data is that the signal that you get isn't necessarily just the activity of the cell that you're looking at. So we compute this ROI signal, which is the sum of the pixels within the cell that we've selected. And then we also compute this neuropil signal, which is where we take all the pixels that are within the masks and ignore those and then compute the average of these surrounding pixels that are not mask pixels, if that kind of makes sense. So that's our neuropil signal. So why do we do that? So when we're imaging our cells, so here's an example cell that's in this XY plane that we're imaging. What's really nice about two-photon calcium imaging is you're doing this raster scanning, and you're very precise at where you're exciting in XY, but your point spread function, so where you're exciting in Z, is actually not so narrow. So this point spread function is quite narrow in XY. You see it's small here, but in Z, it's quite elongated. So when we're exciting this plane in XY, we're also exciting processes above and below the cell that we're imaging. And so these axons above and below the cell are kind of an approximation of them is using this neuropil signal to calculate them. And so if you can look at for example, this neuropil signal for this cell versus the ROI signal. And you can see, so this is the neuropil signal plotted here, the ROI signal, and so you can see how they're correlated. So kind of the background, the cell activity itself is also going up and down with the background, which is this contamination from the neuropil here. But above that contamination, you can see these time points here, which are where the trace is highly active. And so what people do in practice for this neuropil correction is you have this slope here, which is usually around 0.7, and you multiply your neuropil with this 0.7 and subtract it from the ROI signal, and that gives you this, this ROI trace that's been neuropil corrected, in a sense. Does that kind of make sense? So if you do see things in your recordings, for example, like spatial relationships between neurons, you always want to kind of double check things like this, because a neuropil signal is often correlated spatially. Like in visual cortex, you have this retinotopy, and so you'll see the background going up and down with the cells, but the cells are usually less correlated to each other than the background, so you kind of want to check these things to make sure that it's not contaminating your recordings. Okay. And so then you get your fluorescence signal, you subtract 0.7 times the neuropil, and then you should get traces that hopefully look something like this, where you can see these calcium transients that look like this, for example, for this cell, and then some that even look a little more complicated, like multiple spikes riding on top of each other. And so what we can do with these traces is we can deconvolve them, which basically means, so this is an example simulation where we have these spikes. Each of these spikes elicits a fluorescence trace that has this long decay time. So that's what we see in calcium imaging. But we can basically estimate the times of the spikes using these fluorescence traces. And that's often what we'll use in practice, are these estimated spike probabilities rather than using the raw trace, because it enables us to have more temporal precision with the estimation of our signals. Yeah. Yeah, that's a really good question. So it depends on your point spread function, actually. So in most cases on a standard microscope, what we see is around 0.7. But you can do this, you can actually do robust regression and estimate it on a per cell basis if you want to. But it just kind of depends on how much contamination you have above and below versus how much is actually your signal. So if your point spread function is even bigger, your neuropil subtraction coefficient could be even larger. So like we've been using a larger PSF recently, and we use a coefficient of 1, for example. So I think you can check it out empirically and see. Are there any other questions? Yeah. Yeah, that's a good question. So we basically just use a radius of a certain size around each cell, and we just can't remember exactly if it's 150 or something like that. You can also set it in the algorithm to decide how big that radius should be. So you don't want it to be too big, because the neuropil signal is somewhat local. So you don't want it to be the whole field of view, but you can watch the video and kind of see how localized your neuropil looks and kind of scale it based on that, because your zoom level might also be different from our recordings too. Yeah, that's the way we do it in practice, because it's approximately true for the whole field of view, that you'll usually see that the spatial spread of the neuropil is fairly similar across the whole field of view. Yeah, it's a good question. Yeah.
Related to the motion correction part.
Yeah.
So you mentioned you're also able to correct a little bit the Z motion, right? How much is relying on this double plane imaging, and is it possible to do that if you just have one plane?
Oh, yeah, no, sorry, we're not correcting Z movement. This is just diagnosing it. There's no good way to correct Z motion if you only collect one plane, and people should not tell you otherwise, because your cell could basically completely move out of the field of view, exactly like you're saying. So what I've seen in practice people do is our planes are pretty far apart, about 100 microns, so that wouldn't work. But I've seen people do maybe what you're suggesting, like very dense sampling, and then they'll be able to use the planes above and below, and if the cell moves out of the field of view, they'll do that.
I just experienced issues with that. Working on data was
Yeah, I mean, you can also like nan out times where you think the cell isn't present anymore and just say, I can't use that data. Yes, if you see negative deflections, it's a good sign you have Z movement. It's a good point. Yes, I could have mentioned that. Yeah, you're an expert here. Are there any other questions? Yeah. Oh, yeah, no. So basically, in our calcium transients, ideally things look like this. Like you get an input of a signal. We very rarely see that there's like this overall suppression of the signal we're imaging. So if you see this decrease in overall brightness, it's a pretty big red flag that your cell is moving out of your field of view. I've seen some people in zebrafish say they do see some negative signals, but in mice, I've struggled to see it, basically. So if you see negative deflections, you should be wary that you might have movement of your plane in Z. Yeah, that's a really good question. Yeah, so if you're recording for a long period of time and using a lot of laser power, you might see some decay in your signal. So you could do a what people do with DF over F is they'll do a divisive baseline that varies based on the time series. In practice, we kind of try to avoid that because doing normalization like that can be a little noisy on a per cell basis, but that's a potential solution you have. Usually what we do is if we do a recording like that and it looks like that, we lower the laser power the next recording. And that's how we deal with it. And for long recordings, for the Z movement issue, we have in ScanImage, we've developed a plugin, which is available for everyone now for doing Z correction. And so that's what we use to make sure that we can do Z correction during very long recordings, because that is really important, especially for these big microscopes. They just like move on their own. And so you need these kinds of things. Yeah, so when you do the spike deconvolution, one thing I just wanted to add here, particularly with GCAM6, is you see a lot of variability in the spike triggered fluorescence. So we know the time from a ground truth spike, so this is where they do cell attached recordings at the same time as calcium imaging. We can look at the fluorescence aligned to the time of an example ground truth spike that was recorded, and what we see, for example, across these many spikes, is that there's a lot of variability in the overall size of that peak. So in practice, we discourage people from discretizing because we don't think it's very possible with GCAM6, and you actually end up losing information in your traces if you end up discretizing, rather than using all of the information about like what the spike probability is. So that would be our recommendation generally. The last thing I was going to mention was quantifying SNR for your traces that you're looking at. And I think Ben was saying there's a potentially another way to do it that might be available as well. One way that we do it, which is similar to Peter Ruprecht's approach, which was in the Cascade paper, for those of you who are familiar with it, is to compute this noise variance of the cells that we record. So DF here means our fluorescence trace, just additive baseline corrected, basically. And we quantify the noise variance as any variance we have between the difference of time points in our signal. So basically, if we have a nice calcium trace that's nice and smooth, the difference between two frames in that trace should be relatively low. But if there's a lot of photon shot noise, then there's a lot of noise on top of this, then the difference between two time points would be quite high because there's just noise adding on top of it. And so this is an approximate way. So assuming your underlying calcium traces are smooth, this is a way to estimate the noise variance of our cells. And we normalize it by the total variance of the trace. And so these are cells in an example GCAMP8 recording sorted by their fraction of noise variance. So this is a very low noise neuron up to a more high noise neuron up to 0.5. And our median is around 0.27, which we find is pretty reasonable. But this is the sort of thing you can look at in traces that you download from Dandy, and you can check that you really are seeing calcium transients, because you really want to be careful, especially when you're like on the limit, that the things that look like maybe transients are not actually transients, they're just kind of Poisson noise. And here's example traces in GCAM6S, sorted by the fraction of noise variance. So you can see this like beautiful cell here, which is just like all transient, down to a cell that's maybe a little more noisy, but you can still even see transients at this 0.5 level. And the median for our recordings here is around 0.24. All right, So that's kind of the metrics that I wanted to mention when you're looking at data sets. Now I'm going to tell you a little bit about this project on understanding visual processing. And so we're interested in understanding primary visual cortex responses. So for a mouse actually has a really large visual field. So this, you can see, it can see all the way back, even a little bit behind it, and all the way around. Each of the neurons that we record from, they're retinotopically organized, and they'll have a specific receptive field, like for example here, of around a radius of, or a diameter of around 30 degrees of visual angle. And you'll have a bunch of these in an example recording that you do. In the monkey and in us, we have a less large visual field, and we have substantially smaller receptive fields, around 1 to 2 degrees in diameter. But in both species, actually, so in mice and in macaques, our visual cortex, our primary visual cortex, is a large fraction of our cortex, around 10% in mice and 13% in macaques. So we really want to understand like what kinds of models can fit this neural activity, because it is a large fraction of our brain, actually, that's being dedicated to this primary sensory processing. And so to do that, we build predictive models of V1. So you take as input, we have an image that we input to our model that's going to try to predict a V1 neuron. The first step in the model is to have a convolutional layer. So how this works is you have a filter that you run along the image. So this is the input image here. And then you get an output like this green image here, which has activations at each of the input positions that you have here. And so in practice with a convolutional layer, you usually have multiple filters and then what we call these multiple output channels here. And for our example neurons, so like I was saying, they have specific renotopic locations. So we're going to have them pool over activations in a specific part of the visual field. So for example, we have them learn this where they're pooling from, which is like this example circle here. They'll pool across each of these channels. And we'll fit weights for how much they should pool from each of these channels. And so we'll fit this spatial pooling, these weights, and also these filters in our models. And hopefully we fit these with gradient descent. Hopefully they fit pretty well. So here's an example neuron. The activity to many images is shown in black, and the predicted response is in blue. And we fit this simultaneously across many neurons. So each neuron has a neuron-specific readout, and but these filters, convolutional filters, are shared across the whole population. And these are what some example responses look like. Does that make sense? All right. And so one of the first things we wanted to ask with these models, and so this is work done by Fangtong Du in the lab. So she fit these models from images to neural activity. And so we wanted to see how large these models needed to be to explain the neural activity. So we looked at how many convolutional layers we needed. And then we always have these neuron-specific readouts that I explained. So we started with 4 convolutional layers because that was kind of the state of the art in the field. And we found that the explainable variance that, the fraction explainable variance explained that we achieved was around 0.73. So this is normal. I mean, I won't go into the details too much, but basically we can't explain all of the variance in the neural activity. As you'll see in Atika's talk, there's a lot of neural variance related to the behaviors and things like that. But the amount of signal variance that's related to stimulus responses, we're explaining about 3/4 of it with this model, which is substantially higher than previous models because we're showing so many images. So we're showing at least 6 times more images than previous studies have done. And so what we basically now we varied the number of convolutional layers in our model, and we found actually we didn't need 4 in order to explain the neural activity. We really only needed 2. You see the saturation here of the fraction of explainable variance explained at around 2. And our model also outperformed previous models, partly because we have a more expressive readout than neuron-specific readout than these previous models. So we really only needed two convolutional layers to explain these million neurons in mouse V1. How about in monkey V1? So we used a nice data set from the Tolias lab where they showed over 7,000 images to monkeys, and actually also at a fast presentation rate of 17 hertz. and we looked at how many layers we needed to fit this neural activity, and we found that it actually also saturated around 2 convolutional layers. And our model also outperformed the model that they had in their paper, which was a pre-trained VGG model and using the features of those to predict neural activity. And so what we found was that in monkey V1 and mouse V1, we could explain these neural responses with just 2 convolutional layers, basically. Yeah.
Can you explain a little bit the logic of this? Yeah, can you delve a little bit into the logic of this neuron-specific readout? I think I'm curious to know how much neuron-to-neuron variability there is in this convolutional layer. So are there neurons that are best explained by 1 layer, some by 2, some by 3, and then when you average, you kind of get these 2? Or
yeah, that's a good question. So there are some neurons that are better explained by the first layer, but a lot of them need 2 convolutional layers, especially in the mouse case. In the monkey case, there's more of this different distribution, but in the mouse case, you see in the monkey case, basically 2 layers doesn't do much better than 1 layer. But in the mouse case, a lot of these neurons are more complicated, but it is something that you can quantify, like for the complexity of a neuron, like how many nonlinearities do you need to explain it? Yeah.
Thanks.
Yeah. Yeah, there's a question in the back.
Besides the number of layers, you can also change the number of channels in each layer.
Yeah.
If you change the number of channels, you have affect this result.
That's a great question, and that's exactly what we did next. Was we tried to see like what is the minimal size we need and how does that affect performance? Yes, exactly. Because we wanted to see how. So we started with just 192 in every layer, and then we tried to change the size of COM1 and COM2, just having 2 layers. And so in mouse and monkey V1, we looked at the number of COM1 channels versus the number of COM2 channels or filters, and what you see is the columns are kind of all the same, like pretty constant in color. So this is the FEVE, so how much variance we explain, which basically means increasing the size of this first layer doesn't actually really matter very much. But you can see this increase across the rows, which is corresponding to this increase in the size of our second layer. So that actually was important for explaining the neural variance, and we found like an approximate maximum at around 16 convolutional maps in the first layer and around 320 convolutional maps in the second layer for both mouse and monkey V1. And we can visualize, so when we just have 16 in the first convolutional layer, we can visualize these filters. We see on-off filters and then gabors as well in both the mouse and the monkey case, even though these are at very different scales. Like I was saying, the mouse receptive fields are around 30 degrees, the monkeys are around 1 to 2 degrees, but we see similar types of features of these gabors at different sizes. So our first conv layer kind of looks like inputs from LGN and maybe what layer 4 in primary visual cortex is computing. And then the second convolutional layer is really this big expansion of features that we think we need to explain the diverse responses that we see across these million neurons in mouse V1 or 300 million neurons in monkey V1. You need these many diverse features to explain this activity. So we wanted to know if this architecture is useful for tasks. So this approach with this really large second convolutional layer is not actually what happens in practice in computer vision. Yeah, sorry. Yeah.
But I guess I was wondering, can you have a lot of everything layers, but a deep number? Then you can also explain V1.
Yes, I think that's a very good point. And so we could have even like had many layers and even a smaller receptive field in the first layer and explain V1 because these are like kind of like universal function approximators. So we can kind of get whatever we want out of them. So it is a good point. But we are kind of assuming that ideally the minimal model is going to be closest to maybe what the brain is doing. So that's the assumption we're making here, and then we're trying to find that minimal model. But it still needs to be mapped to the biology, for sure, to know that that's actually the way that's happening. So in this case, we have 3 nonlinearities, basically, for each neuron, although you could have more and change things if you want to. Yeah.
I have a question on your how you did the first the 2 network, so two layer argument and then the size of the layer argument. So did you do that independently, or did you also explore this space of all combinations of layers and sizes of layers?
That's a good question. So we did play a little bit of having 3 conv layers and making it bigger to check we couldn't do better. But basically, in this case, we always had 192 in each layer, which is very big. So we're kind of like, we couldn't get more performance gains in 3 convolutional layers, for example, if we made it even bigger. So we did try things like that. What happens also when you make them deeper is you end up actually having more overfitting of these neurons, which maybe is an argument for the simpler models as well. But yeah. Good questions. Are there any other questions? Okay, so we wanted to see what this architecture might be useful for with this very large second layer, which has many diverse features. And so we trained this neural network with these 2 convolutional layers to perform texture classification with these different images of like leaves and rocks and things like that. And what we found was that if we look at the performance, the accuracy on this task as a function of the size of conv1 and conv2, we see that the accuracy doesn't change very much across the columns like we saw before. It's pretty flat. It really is having a wide second layer that helps with a task like this texture classification. And we see a similar thing if we train this neural network to do ImageNet. In this case, we had to add a fully connected layer here to improve performance because ImageNet is harder. But basically, on a downsampled version of ImageNet, we get around 40% accuracy, and that requires having many filters in the second convolutional layer. So this network, V1 seems capable of doing these different tasks. So we wanted to know if mice can also perform these kinds of tasks like texture discrimination and potentially learn these visual memories of texture while they're doing these tasks. And so Lin Zhang in the lab, he trained mice to do visual texture discrimination. He had a circle corridor and a leaf corridor. In each of these corridors, a random sound cue is played, at a random time. And in the case of the leaf corridor, this random sound cue is followed by a water reward. So he trains these mice for 2 weeks to do the task. This is what it looks like. Here's a mouse. So you can see the mouse is head fixed here for people who haven't seen mice like this before. They run very fast. This is in real time. This is a rewarding corridor, so you're going to see the mouse stop now, and it's collecting the reward. It heard the sound cue, and then it was licking, and it collected the reward. Here's another rewarded corridor. It should also stop here. See, it's collecting the reward. You see it was at a different time because we have random times of the rewards, which is helpful for doing, for kind of distinguishing reward-related signals versus visual signals. And then it just ran straight through that corridor, which was non-rewarding. And so Lin trains these mice for 2 weeks. Before learning, they lick pretty similarly before the rewards or before the sound cue in both corridors. And then after learning, the mice lick substantially more in the leaf corridor compared to the circle corridor, so they can clearly learn this texture discrimination task. We also made this task even harder for them by matching the Fourier spectrum of these images. So they have to use higher level features in order to do the task. And what we also did to try to understand these representations, so we had task mice that were trained in this task with water reward in the leaf corridor, and then we had another set of mice that ran through these same corridors, had these sound cues, but were never given rewards. And so we can compare representations between these task mice and these unsupervised mice as well. So to look at these, the representations, we looked first at the stimulus-selective neurons and where they might be in the brain before and after the mouse learns this task. So we defined stimulus selectivity using a D' index, basically the difference between the responses, the average response on the 2 corridors divided by the standard deviation of the responses across those trials. And we can look at the responses of the neurons on the circle and leaf trials. So here's an example leaf selective neuron. It's leaf selective neurons. Their example activity on the leaf trials, you can see is substantially higher than the circle trials, and they have specific positions they're tuned for. And we see also example circle 1 selective neurons that are high on circle trials and low on leaf trials. And so now we wanted to look at where these neurons are in the brain before and after learning. And so We look at neurons with an absolute value of D prime greater than 0.3, which means basically the leaf selective and the circle selective neurons. And so what we found in the task mice that are trained on with the leaf corridor rewarding for 2 weeks, so there's a lot of selective neurons. These, what's purple here is primary visual cortex before learning. But then after learning, we see this large increase in the number of selective neurons in these higher order visual areas, these medial areas that are close to retrosplenial cortex. And so the next thing we wanted to ask was, is this because of this task learning, because they were learning that leaf is rewarding or circle rewarding, or is it due to the visual experience of the mice running through these corridors for 2 weeks? And so we compare these to these unsupervised mice that never got water rewards, were never water restricted. And actually we saw quite similar changes, particularly in the medial area, that there was this big increase in the number of selective neurons in this area. So what this suggests is that these changes in these higher order visual areas are not related to the perceptual learning or learning of the valence of 2 corridors, but actually just due to running through these corridors. Sort of like you walking around Janelia and learning landmarks in the corridors, like you don't need to get a sip of coffee or reward every step you take to learn the features of your environment. Okay, so we can summarize this. Basically, the average percentage of selective neurons in V1 is pretty flat for both the supervised and, unsupervised and task mice in green and purple. But we really see this big increase in the percentage of selective neurons in both the task mice and the unsupervised mice here, suggesting that this is a part of the brain that's potentially useful for unsupervised learning, and we're thinking of basically as a place for storing potentially visual memories in the brain in a compressed way. We also looked at lateral and anterior brain areas. These anterior areas up here are some of them are kind of part of mouse posterior parietal cortex, which is more related to reward signaling, and we do indeed see this increase in selective neurons there in the task mice, but not in the unsupervised mice, suggesting that these neurons are related to task learning. And so I'll quickly show you these task-related neurons, and then I'll wrap up and we can go to FaceMap. But basically, and one more quick thing about these unsupervised neurons. So what Lynn did after he did this leaf 1 task, he showed this new exemplar of leaf 2, which was not rewarding. And in the first session, the mice also licked to leaf 2, so they licked to leaf 1 and leaf 2. In the unsupervised case, they're never licking. They just observe leaf 1 and leaf 2. And so before learning, the first time they see leaf 2, the representations of neurons in these higher order visual areas are quite similar to leaf 1 and leaf 2. But actually, after learning, we see this orthogonalization of these patterns in both the unsupervised and supervised mice. So basically, these patterns are visually similar, so they drive these neurons similarly that are visually responsive, but then they actually get pushed apart, which we think is a potentially some form of contrastive learning that's actually happening in this higher order brain area. And there's more details in the paper if you're interested in looking into that. Yeah.
Thank you for the intro. I really enjoyed reading this paper when it was on our.
Oh, thanks.
Yeah, but there was a question that always intrigues me, because even in the medial area, we see that there's a difference in the task mice and the unsupervised mice. Do you make any information about that difference, that the blue bar is still higher than the purple bar after the learning?
It's a very good question. Our hunch would be maybe it's still like engagement related, that the task mice are even still more engaged in the environment would be our hunch. Because, for example, we've tried doing these experiments in open loop where the corridors aren't locked to the mouses running, and it doesn't work so well. So I think the engagement in the task is important, and the task mice are even more engaged than the unsupervised mice would be my hunch. But we still see these changes in unsupervised mice, so we are pushing on that, and it's nice not to have to train mice. So, yeah, it's a good question. Are there any other questions? Okay. All right. Yeah, I basically gave you a whirlwind tour, but there's more details in the paper here. Maybe I'll quickly show this. Basically, we also found a substrate for supervised learning in these higher order visual areas. Basically, we found these neurons that seem to ramp up to the time of the reward, which is this green time here. So these are neurons by time, sorted by raster map. I think some of you have used raster map this week as well. So basically, you can see these sequences of neurons in these two corridors, in the leaf and circle corridors, at these times in blue and red. And then we see in the leaf corridor, which is rewarding, these neurons that ramp up to the time of the reward and seem to turn off after the reward is delivered. And basically, after the mouse consumes the reward, there's no more reward in that corridor any longer. So the reward prediction or reward anticipation goes to 0. And so we see these neurons. I'll zoom in on them, basically ramping up and turning off at the time of the reward. They seem to be in these anterior, higher order visual areas. And we can summarize them by sorting these trials by the time of the sound cue. We don't see a lot of activity in this neural population in the circle trials, but we do see activity in the leaf trials that seems to ramp up to the time of the sound cue and then shut off after the time of the sound cue. And so basically what I tried to show you here was that V1 is kind of this large, diverse set of feature extractors. There's more information in the paper, and all of the code and the data is available, so you could fit these neural models if you want to your own data or try out our data. And then in the case of Lin Zhong's study, also all the data is available. And I showed you that there's this flexibility in these representations in higher order visual areas. And if you check out the paper, you'll see how unsupervised learning improves supervised learning. And so now we'll do a switch off. You're ready to go? Thanks, everybody.
Do I just connect this?
Yeah, you can. Yes, but we need to put this in here.
Oh, HDMI. I was wondering if it was advanced.
Yeah, and then this is this. So if you want to use this, you can.
Yeah, it's a good question. So in the medial area, we also found 2 layers was sufficient. That's not published yet, but yeah, so we have tried in other brain areas too.
Hi, everyone. So I'll be presenting on FaceMap. And first, I'd like to start off by talking some of the previous work that led to the development of FaceMap. And specifically this study by Carsen and others, where they recorded activity of 10,000 neurons in the mouse visual cortex. This was done using 2-photon calcium imaging and dual plane imaging, as you can see here. And simultaneously, they recorded behavioral videos of the mouse using multiple cameras that capture the animal running on the ball and its other body and orofacial movements. So the mouse was head fixed and it was in complete darkness, so there's no visual input coming in while we're imaging in the visual cortex. So here you can see activity of the 10,000 neurons I showed, which now you can extract spikes using Sui2P and activities sorted using raster map, which many of you have been using. And in the paper, they asked the question, when there is no visual input, then what is driving this spontaneous neural activity? And is it the mouse behavior or running or orofacial movements that they recorded? So to study this, they performed PCA on the behavioral videos, as you can see here, to get these components that represent or capture motion of the mouse face. And then using a linear model to predict this neural activity from these behavioral PCs, you can see that the predicted neural activity looks very similar to the recorded activity, meaning when you quantify this, a large fraction of this activity, spontaneous activity, was explained by the spontaneous behavior of the mouse. But there are a few challenges when using motion PCs for studying neural representations. So as you can see, they lack interpretability, as you can't tell what behaviors are relevant for those neural representations. And second, the linear model that was used did not have temporal information, so we might be missing out on some of the temporal dynamics of behavior that are relevant for neural activity. So we decided to use a different approach that some of the labs had also been using for quantifying animal behavior, which is animal pose estimation, where you can draw these key points or label key points on the mouse face to specifically track different regions such as the whiskers, nose, eyes, and mouth. And so we turned to existing tools such as Deep Lab Cut and Sleep, but these tools often require training a different model for multiple different experimental setups, so they lack specialization for our problem. And we wanted to create a tool that's just specific for the mouse face and that can capture these movements from various different camera angles. So we developed FaceMap, which is a smaller network compared to Deep Lab Cut and Sleep. I won't go describe all the details, but I'll say it's a UNet-style neural network with skip connections. And we train it on 2,400 frames from multiple mice collected from different camera angles, such as a side view and top view of the mouse face, and hoping to create a generalist model so we can get videos from different setups and extract our key points. And here you can see the performance on a test mouse and the key point tracking in real time. So now, having our key point tracker, we can now quantify these various orofacial behaviors, such as when the mouse blinks, we can look at the different positions of the eye key points, or when it's whisking, you'll see these oscillatory-like movements in the traces, and sniffing behavior as well. So next, we compared FaceMap Tracker with other state-of-the-art pose estimation tools, like I mentioned, Deep Lab Cut and Sleep. And for this comparison, we used two different metrics. One, we're interested in how precise or accurate the model is. So on the y-axis represents the error. So lower the error, the better. And secondly, we're also interested in how fast you can process a single frame in our model, ideally using it for closed-loop behavioral experiments for real-time analysis. So our model stands here, and as you can see, it's not only just as accurate as the existing models, but we're able to push this boundary of the inference speed or how fast the model is by making a much smaller network specific for the face. Yes?
Do you have anything with tongues? Like when you lick?
Currently, there is no key point for tongue, and it depends. I know some labs use like a sensor to detect licking time. There is a trick to the model, which I suggest people. There are other key points for a paw or something, and you can retrain a few frames by like calling that tongue. But that's one of the ways to go about it. But there's like...
It's also difficult because the tongue like appears and disappears.
Yeah.
Yeah.
On a side project, if you're interested, there is a paper that uses a unit model for doing mass segmentation of the tongue from the side or the bottom view because they want to get a 3D reconstruction of the tongue, which is useful if you're interested in like the volume of the tongue and, like you said, protracts, retracts.
Kind of follow-up question to that. How did you pick the key points that you wanted to track?
Yeah, so by just first like staring at a lot of these mouse face videos, then you want to look at which whiskers are easily identifiable. So from like barrel cortex literature, you'll see like all these whiskers map onto specific positions in the barrel cortex. And also, if you were to observe these, like the few whiskers in the first few columns were much more noticeable across multiple videos. So I just picked 3 key points. You could do more, but turns out they're very correlated at the base, about 99% correlation, so you could even use a centroid of these key points. So that was for the decision for whisking, and for eye, the only things we can capture is blinking, and I guess if there's some movement back and forth that these key points around. So anything that's easily identifiable across mice. There were other key points that we started off with, which was like somewhere down here and here, but then it's not reliable. So many different faces of mouse, and you get a lot of noise in human labeling as well. So these key points are easily identified, like the tip, the bottom of the nose, and the top, and they do happen to also capture when the nose moves left and right, up and down. Yeah.
And
Yeah, so the previous version of FaceMap that Carsen had still does the pupil size tracking. Maybe I don't have a video here, but basically you draw an ROI around the pupil, change the saturation. So we'll have like a circular elliptical ROI and then track the center of mass of that position. That's also something we've been doing more for my current project, which I won't cover today, but yeah, you can check out FaceMap for that.
These are in total darkness, so you can't really see the people area, but yeah, we have some cool things. We track people in darkness.
Yes.
Can you explain what the two outputs there?
Oh, yeah. So we used a similar objective as Deep Lab Cut does and some other tools, which is our prediction is one of them is a heat map, which is a probability where a key point lies. And as you can see that we're not scaling our output map back to the original frame size, which is you have to get the actual positions. So what we do is we store these dxdy or like the differences in position in the original frame, and so these are called location refinement maps. And you use a so this is a this one's classification loss for the heat map or probabilities, and the other one is mean square error loss on the actual position. So we combine both information, get the maximum of where likelihood where the key point is, and then use the information on the location refinement to map it onto the full image space. Yeah.
So yeah, since we're asking questions, I have a question from a few slides ago.
Okay.
I think from the very first slide or second, where you do the prediction with the raster map.
Yeah.
Yeah, so I didn't really understand that very well. I mean, so what is the prediction based on? I mean, what is the data that is fed into the...
So this here exactly. So the video PCs,
Yeah.
which is 500 components, are our input to the model, and our output, because it was much larger recording, was 128 PCs of the neural activity.
But like, okay, so the raster that's on top is actual neural activity, right?
Yes, that's
So
the
500
yeah,
different
and this is the test data, which is prediction from these behavioral PCs during that time period.
Okay, but like, I mean, is the... So, I mean, is the prediction completely independent from observed neural activity? So I'm a bit fuzzy on that.
So the model we use is reduce rank regression.
Yeah.
And so you will have your training set where you feed in your behavioral data, and your Y is your neural activity output, all during train times. And then we can feed in the behavioral data X during the test time and get this prediction.
Okay, so when you said that, so why does this show that the actual neural activity is explained by the, I mean, isn't like the model sort of necessarily trying to fit it?
It didn't have to work though on it. There's plenty of, like you could, if we did this just, for example, to running speed, this would look much worse.
Okay.
Yeah, so I'll have a plot later on where we'll compare these predictions of raster maps. And by when I mentioned explain, I'll go into when I meant it's a variance explained computation where you can quantify how much of this variance you can explain. So, okay, so we'll get to that in a few slides. But first, I'll also now go back to the point that we want to use it for different experimental setups. So we asked labs to contribute their behavioral videos and test how our trained model on 2,400 images performs. And for the different labs, you can see that on some of the key points, it works well, while failing on some others. And this can be due to changes in lighting, such as brightness or saturation. So we added a feature where users can correct some of the key points that are incorrect and leave the others unchanged and feed it back into the model and retrain. So through this fine-tuning process, we can see that our fine-tuned model's performance reaches near optimal performance around 10 refined frames. And you can see those predictions here. So this process takes less than 2 minutes because it will already generate key points, and you just have to move it around to its right location and then retrain it. So now that we have a model for precisely tracking and quantifying these orofacial behaviors, but we're still interested in how these key points relate to neural activity. So we'll go back to, like I mentioned, previously we used a linear model, but now we want to develop a nonlinear deep learning model that can do end-to-end fitting of these key points to neural activity. So I'll briefly describe the details as to how we came about this model. So like I mentioned, the previous linear model did not have temporal information. So in this case, we feed in our key points and they get passed through these 1D temporal convolutions, hoping to capture those temporal dynamics of behaviors. And then to further push this boundary of how much activity we can explain from behavior, we added nonlinearities to the model and also a couple of hidden layers, hoping to get a low-dimensional representation, which can then be projected back onto or predict our neural PCs or prediction output. And in this case, yes, we're using PCs because if you have a large data set or if you have less than 200 or something neurons, you can just predict the single neuron activity itself. So we'll go back to the raster map I had shown earlier. Now this is our raw visual cortex activity recorded in about 35,000 neurons, and this is test data. And again, the mouse was head fixed in complete darkness, so there's no visual input. And now I'll plot what does the output of our deep learning model looks like. Over here, this is a prediction from our network. And on the bottom here is a prediction from our previous linear model that I had showed you. So you can see that the deep learning model can capture some of the finer features of the neural activity, which we don't see in the model down here. And now to quantify, I'm going to plot the variance explained in our test data for multiple mice recorded in either sensory motor region or the visual cortex. And comparing our linear model to our deep network, you can see that it almost doubles the amount of variance explained in both sensory motor and the visual cortex.
So the main idea here is that if the neural data could not be explained from the motion, the model would not be able to generate such a prediction, right? I mean,
yes, you could feed in random noise as control, and like Carsen mentioned, running speed, just like one variable, or pupil size, and the prediction would look worse than even this one. And this currently is at about 10 to 20%.
Okay, thank you.
So now coming back to one of the points I had mentioned about interpretability, we wanted to, I'll go briefly through these slides because I don't know how much time I have. So we wanted to study the low dimensional features from our model. And to do that, we extracted these deep key point features and fit it in this hidden Markov model with 50 discrete states, and to study the different behavioral states and their dynamics. And here on the top, you can see a short time segment of the mouse going through the different states or inferred states. And we can also plot the activity of neurons by selecting the top neurons that were most selective or driven during each state. So state 1, and then each row here represents a trial or every time that state occurred, and the average activity of those selective neurons at that time. And the black line represents the state onset, start time, and end time. So you can see some states were shorter and others were longer. So we can look at what did the mouse behavior look like during each of these different states. I'll show you just a few examples where there's one state, and you can see that during this time period, most of like all the different trials, you can observe the mouse was grooming, and in another we observed this behavior of blinking and running or resting. So these were some of the easily identifiable states that you can call say what the mouse is doing, and there are some other ones too that may be more subtle differences or harder to notice. So yeah, I think I'll summarize here to say that we hopefully developed a fast and accurate mouse or facial key points tracker and a new encoding model that can do this end-to-end fitting from key points to neural activity that also helped us identify behavioral states. And we shared our work in a GUI that you can install from GitHub. So what you can do is load in your behavioral video, mouse video here, and then run key points and get the key points traces at the bottom here, or also the motion PCs that I mentioned earlier. And yes, it has other features where you can add ROI for people, and on the right you'll see the pupil position and get traces for that as well. So yes, this is available on GitHub, and happy to answer any more questions. Thanks. Yeah.
It's a bit of an extension, but there was this paper from Atika about like facial expressions and emotions. But have you tried any of this stuff?
We have not run there. I have definitely read the paper, but no, have not tried that approach. It was like the histogram of hogs, what they call, and then you do clustering and see if there are different clusters represent different states. Yeah, that's.
I was just curious, like how this could generalize approach if you would have tried.
Yeah, here this was built more specifically for because even if you weren't to do the prediction to neural activity, there's a lot of behaviors you can study just from the key points information themselves, which again, with their approach of using hogs is similar to using motion PCs or movie PCs, which is harder to interpret or do something with it.
Thank you for a very nice talk. So I was thinking about the relationship between the PC approach and this the feature extraction that you did. If I understand correctly, it's related to capturing the spatiotemporal components of activity compared to just the spatial components. And something that, I also remembered of a result that I think is from the Charchland lab, where they fitted neuronal activity using videos from mouse face. And something that surprised me back in the days is that it seemed like this it's the air, like this very peripheral air, that explained a lot of the variance of neuronal activity. And I was wondering, so first, have you compared this like your approach versus the PC approach, that even if it doesn't have the full temporal dynamics, still captures the whole video? And second, it seems like it's doing a very good job, like the tool with these few key points. So will the idea be that, all of these other air movements is probably just correlated with the key points that you already have, and so it's not really adding any?
Yeah. Those are very good questions because there's more to this paper that I didn't go into. But to the first point of your question, comparing to the PC approach, so the second part of this plot, this is just using key points in the linear model versus our deep network. Then we also have the movie PCs comparison and the motion PCs. And those lines look very similar in trend as our key points, which is good because it tells us that we are explaining as much of the variance that you can explain using the whole face of the mouse itself. But then there's the advantage of using key points because our movie pieces are 500 principal components compared to these 15 or 13 distinct key points and their positions. And your other question was...
I guess it's related and yeah.
But yeah, we've also done comparison of... because now we can separate different parts of the face, whiskers, nose, and eye. So you can see how much each different regions predict neural activity. Turns out, yes, not as good as using all those different regions, and often whisker and nose better than other parts.
So it's basically highly redundant, all the motion you have all over the whole face, and then, yeah, with these few markers, you can capture most of the, like, whole...
Yeah, because as you'll see, like the whisker pad itself, like moves very, like the whiskers on it are very correlated. Maybe there's more information at the tips or other stuff happening, but at the base it's very correlated, and often nose is very correlated to whisker, but there's still different information you can get from a different view of the nose, which is something, yeah, we have been currently looking at. So hopefully in the future you'll see more of that.
Thanks.
Mm-hmm. Yeah.
From this plot, I see some there's decent variation, but it's also explained by visual and vertical activity. 30%. I wonder if that's considered as kind of noise or overfitting, or if there's, because it's recording the dark.
Yes.
Does it consider as maybe vertical signature behavior, or maybe more noise or normal? I don't know how you want to explain these predictions.
So why is the prediction so good in the visual cortex when there is no visual input coming in? Well, so far, what we explained here, that it's related to those behaviors. It's very likely they're related to other indirect ways of neuromodulation, arousal states, and those behaviors that are linked to those processes. But that's the extent of explanation we have so far, that that activity can be reliably explained across mice using these behaviors. Yeah. Yes.
There is some work from Jonathan Wicklow. They do that 3D tracking in the rats, I think. And they see that as you go more front to the cortex, you explain more like facial, oral facial, head-related, like movement. And here I couldn't, like, you see this increase of normalized variance explained, like, as you go a bit more frontal with the sensory motor. Doing something related to that, like a little bit of like localization, like better explain like facial movement as you go more to the front of the cortex. Or like this.
Yeah, for this study, we only looked at the sensory motor region and the visual cortex. And within this window, we did do some analysis. I can show you a slide on it, which is where we had a question that, so here I'll show, we have the sensory motor window and each point here represents a neuron. And what we did is like have the entire population divided into subpopulations using k-means clustering. So these are 100 clusters. And so each of those clusters ideally should have similar firing patterns. So we were interested in the question, like, is it, like you mentioned, is it certain region of the sensory motor area that's more related to behavior, or which of these clusters? So you can see that some of these clusters are more spread across and some are more localized based on their firing pattern. And then what I can do is I can plot all those subpopulations and color it by different colors. And then on y-axis, we can plot how for each of those clusters, how well are they predicted by neural activity on the y-axis. And on the x-axis, we have the locality index, meaning how localized, like the gray one, meaning how clustered it is. So the more dense, then the higher locality index, more spread across, lower locality index. And as you can see, we see this trend that the clusters or neural subpopulations that are better explained by behavior are actually more spread across the sensory motor region. And also in the visual cortex, we observe similar trend compared to like basically no spatial localization of representation of these behaviors. But yeah, fun paper. I'll make sure to check it out. Thanks. Okay, thank you.